Hi, everyone. Welcome to Foresight's Biotech and Health Extension Seminar Group. And welcome to Michael Levin. Once more, we have you on. I think it's now the third time that we have you on for a seminar, at least. We've had the pleasure of welcoming many of your grad students and other collaborators into various other Foresight venues. And I think it's safe to say that there's very few people that are so versatile, so at the frontier of many fields, connecting them in such rich ways as you are. And so usually we don't just talk about biotech here. It usually goes into some Agential stuff, into some AI-related bits, into some regeneration stuff, but also into some xenobot stuff. And so let's see where this conversation leads us. But thank you so much for coming back. You've been in high demand here, and I hope it's not the last time. But for now, I'll just hand it over to you. I'm going to share more info about you in the chat. Thank you. The stage is yours.
Excellent. Yeah, thank you so much. I'm delighted to be here. Thank you for this opportunity. As you said, I've been here twice, and I was trying to think what I should show this time that you all haven't seen before. I've made an entirely new talk this time that I've never had, so you're all guinea pigs. So let's see how this works out. It's got some new things. Just in general, as always, the primary papers, data sets, software, everything can be downloaded here. All the peer-reviewed stuff is here. And then this is my own more personal take on what I think these things mean in case anybody's interested. But all the reprints and everything else is here. Okay. I think that here are the main points that I'd like to get across today. Basically, I want to talk about the living material as an agential material. And I think that it's not quite what we think it is, which I think has implications for biomedicine and beyond. So the first thing I want to talk about for anybody who hasn't seen some of these ideas is the emerging field of diverse intelligence, the effort to communicate with unconventional minds. And so these are different kinds of agents that are not the typical brainy mammals that most people deal with. And in particular, the idea of emergent cognition, not just emergent complexity. So emergent complexity is easy. It's quite simple to get complex, unpredictable behavior out of simple rules, but that is not what I'm talking about here. I'm talking about a degree of actual emergent cognition, intelligence, problem solving, and I'll define some of these things. The big new thing that I want to talk about today comes from a paper that just came out a couple of weeks ago that has to do with the way that I think biological agents interpret and reinterpret information on the fly. And in particular, I'll talk about this bow tie architecture and the importance of creative confabulation. And then I will also talk about the usual kind of biomedical implications of thinking beyond bottom-up emergence. So I think the whole thing could be summarized by saying that living bodies have a kind of multi-scale competency architecture, which illustrates really remarkable plasticity and problem-solving skills beyond the things that it was specifically evolved to do. And I hypothesize that this all boils down to something that I will describe as the paradox of change, which forces living systems at multiple scales to be active sense-making agents. And I think this has massive implications for medicine. So I'm going to show you a couple of examples of what I mean by these problem-solving kinds of capacities. So first of all, let's just consider a planarian. So this is a flatworm. You'll see some more of these later. They have the ability to regenerate all of their body parts, including their heads. And we did the following experiment, which is detailed in this paper. If you put a planarian in a solution of barium, the barium is a non-specific potassium channel blocker. What will happen is that they literally lose their head. So their heads explode because they're full of neurons and other cells that really need potassium flux with the outside medium to be happy and to survive. So their heads explode. But remarkably, if you leave them in the barium, then over the next couple of weeks, they regrow a new head, and the new head is completely barium adapted. No problem. Now, what's interesting about this is that planaria are normally not exposed to barium. And so just think about what this means. I often visualize this as a kind of a nuclear reactor control room where every knob is a separate gene. So you've got, 20,000 different steps that you can take in transcriptional space. You're under a massive stressor. Your head just exploded. You're in this physiological condition that you've never experienced before. Which of your genes should you up and down regulate to solve this problem? You don't have time to try them all combinatorially. This is not a situation like in bacteria where everything will die except the one rare survivor and he repopulates the population. Planarian cells don't proliferate that fast. So you really have to solve this problem. You have to figure out which of your effectors in transcriptional space are going to help you. And it turns out that they zero in on a very small number of genes. So we did transcriptomics comparing these barium-adapted heads to naive Planarian heads. There's actually a pretty small number of genes that allow them to do their business in the absence of potassium flow, which is fairly remarkable. And so the question is, how did they know which of those genes to deploy against this problem? So this is an instance of the more general thing we call intelligence, which is use the tools that you have in novel ways to solve a problem you haven't seen before. So one thing that's interesting about this is that for biomedicine, and I'll sprinkle these throughout the talk, like what could the implications be? One interesting thing is that if we understood how they were doing it, or barring that, if we were able to communicate with the cells as a kind of in vivo simulation or an avatar of some sort, and ask for a given stressor, for a given situation, what would you do to resolve it? It seems to me that we have now the beginnings of this idea that these cells can provide solutions to things we otherwise would have had no idea how to do. And if they can do this for barium, maybe they can do this for lots of other conditions. I will also just parenthetically note that this is the beginning of what you really need if you were going to do some kind of evolution that was not random with respect to its outcome. So typically the reason that I used to think that the reason that hard Lamarckian inheritance doesn't work is because once you know what your phenotype is, development is not reversible. You can't compute it backwards. So how do you know which genes for complex phenotypes? Let's say you're a giraffe, you want the longer neck. There is no single gene for a long neck. How do you know what to mutate? You have no idea what to mutate. And so you end up with the standard picture of Darwinian evolution where you have random mutations in selection. But it now turns out that if it's true that cells actually have some kind of mechanism doesn't even have to be perfect, it just needs to be better than chance of zeroing in on which loci are relevant to the problem that you're facing. Now mutating those is pretty easy with CRISPR systems and whatever. That's the easy part. The hard part is knowing what to change. And it may be that these cells know what to change. So I think that's incredibly interesting. And I think we need to understand how they do this. Now, within cells, you have molecular networks, and these, let's just take the example of a gene regulatory network or a protein pathway or something like that. You look at you look at the models, and they are ordinary differential equations tying together some number of nodes that up and down regulate each other. And it looks like a pretty stupid system in the sense that it's completely deterministic. It's quite transparent. All it is some chemicals turning each other on and off. But if you're willing to test its competencies, you can get surprises. And so one thing we did was to basically treat these simulated networks as animals and do different kinds of training on it. So the training is you pick a node and you call that one of your stimulus nodes, let's say the conditioned stimulus. You pick another node and you use its value as the response. And you know that there's some node which is the unconditioned stimulus that when you trigger this, the response comes on. If you trigger this one, the response does not come on. So this is an example of Pavlovian conditioning, right? You've got the meat, you've got the bell, and you've got the salivation response. So it turns out that if you look at real biological networks, not random networks don't tend to do this, but real biological networks tend to have 6 different kinds of learning that they can do. They can do habituation, they can do sensitization, they can do associative learning, this kind of Pavlovian conditioning. And what happens after you present these stimuli together is that just triggering the conditioned stimulus alone, which used to do nothing, but now having been presented together, now it is sufficient to activate the R. So the network has learned in this behavioral context. So this is present inside of cells. In fact, there's lots of these networks inside of cells. That's all described here. And that means that now on the table are all kinds of interesting things like drug conditioning and associative learning and other ways to use pulse stimulation of drugs. And we are now in the process of testing all of this in real cells. This is some of the devices that we've built to actually train cells in this kind of way. So you can see now some of the machinery that might underlie these kind of large-scale problem-solving capacities. There's already learning here. The smallest network that does associative conditioning is 4 nodes. So it really doesn't take much. You don't need a cell, you don't need a nucleus, you don't need any of that machinery. Four interacting genes alone in the right architecture already give you associative conditioning. Now, the problem solving really takes off when you consider the problems in anatomical morphospace. So not just the emergent complexity of going from an embryo to a whole body, but the ability to reach the same goal by different means. So when I talked last time, I talked about this idea of testing the competencies of these anatomical homeostasis systems to get to the same outcome despite the various weird things that you can do to them. So in embryos, you can cut them into pieces and you don't get half embryos, you get perfectly normal monozygotic twins, triplets, and so on. In some organisms, such as in these axolotls, you can amputate anywhere along the length. You get very rapid proliferation and morphogenesis, and then it stops. And it stops exactly when it's reached the correct region of anatomical space, not before, not after. So there's a third kind of interesting example where if you graft a tail onto the flank of a salamander like this, what will happen over time is that this thing will actually remodel into a limb. So this tail up here, the piece of the tip of the tail, which is locally perfectly fine, it's sitting at the end of a tail, and yet the whole thing gets remodeled, including that tip, it becomes fingers, into a structure more appropriate to its global positioning. And so we can imagine that if we understood all of this, the ability to get to your goals despite very unusual circumstances, such as this, a tail sitting in the flank, then this is very useful for regenerative medicine because we could take advantage of this plasticity in patients. Now, the first two look pretty easy to explain by evolutionary forces. This obviously happens during injury. Axolotls bite off each other's legs all the time. This, you can also imagine, okay, sometimes embryos will get shaken apart or whatever. This seems different. This does not happen in nature without the intervention of some sort of surgeon. So why is it able to do that? And that's one of the most interesting things. There are some of these capacities that really seem unexpected. Like here, this is a tadpole. Here's the mouth, here are the nostrils, here's the brain, the gut. And what you'll notice is that it's missing its two primary eyes, but it has an eye on its tail. And these eyes, first of all, the way this was made is by grafting some eye precursor cells onto the tail. First of all, they assemble a perfectly good eye, even though they're in a weird location sitting next to muscle, no problem. Second, when they put out an optic nerve, they put out this one optic nerve here. It sometimes synapses on the spinal cord, sometimes on the gut, sometimes nowhere at all. It does not go to the brain. And yet, if you test these animals, this is a machine that we built for automating the training and testing of these guys. If you test them on visual cues, they behave and they learn. They can see quite well. These animals can see. So now the question is, you've changed drastically the sensory motor architecture here. We've got the eye back here. So these animals with a radically different sensory motor architecture can learn in visual assays, and they can see out of these eyes. So that's weird. Why doesn't it require new rounds of new generations of selection and all that? Why is this plasticity out of the box? The very first time you do this, already they can see. And so this, of course, has biomedical implications too, in terms of sensory and effector substitution, augmentation, and so on. The plasticity is quite large. Okay, so what we have so far is that the biological material, it can solve problems in the sense of responding to novel scenarios. It can do this at a physiological level, transcriptional level, anatomical level, and sort of functional behavioral level. But there's more. One interesting capacity that this material has is that memories can move within the cellular collective. And so the way we know that is that you take these flatworms, and this was something that was discovered by McConnell in the 60s, but a lot of people didn't believe him. So we reproduced all with an automated, that machine that I showed you a minute ago. So what you do is you take these planaria, you train them to eat on a particular surface. So there's you can't really see this, but there's little bumps on this arena that are laser etched, and you feed them on that surface. Then you cut off their head, which contains their centralized brain. The tail sits there doing nothing. And then after about 8 or 10 days, it regenerates a new brain. And then you start to see that it will go look for food at that same location. So what you have here is learning that apparently keeps this information throughout the body in some form that we don't yet know. But what we do know is that when you grow a new brain, which is what controls behavior in this animal, the information from the rest of the body is imprinted onto the new brain. So it's not just that the information is stored everywhere, but it actually is able to be imprinted onto a completely new, fresh brain in some fashion. So this has applications for biomedicine, because if you think about the obvious implication is the stem cell therapeutics in the brain. So if you have a patient that has, 60 years of memories and personality and whatever, and you replace a part of their brain with a progeny of new stem cells, as I'm sure at some point we will, how do we make sure that we're not erasing his personal identity? And it looks to be that, at least in this species, that should be perfect. That's perfectly possible. There are other implications for this potentially. So this is not a strong data set yet, but there have been reports of personality changes in heart-lung transplants that in some way mimic the personality traits of the donor. And again, this is not a clear thing. There's very little data on this still, but it is not crazy that tissue imprint information across the nervous system and onto a new brain. So who knows, right? I don't think that's impossible, and I think we should keep an eye on it. But there's one more feature of this, which I think is even more significant, which is that the idea here is not just to hold on to the memory, and it is not just that you store it elsewhere, you hold on to it really carefully, and then you imprint it onto the new brain. It's more than that. It's not just the fidelity, because the other instance of memory moving across bodies is this. Imagine a caterpillar. A caterpillar has a specific brain designed to help this soft-bodied creature navigate a two-dimensional world of leaves and eat those leaves and so on. This has to turn into a moth or a butterfly, completely different, hard. You need a controller to drive a hard-bodied kind of architecture. You need to fly, so you now live in a three-dimensional world. You no longer care about leaves, you drink nectar. And so during this process, the brain is basically taken apart. A lot of the cells die, all the connections are broken, everything is completely refactored. You get a new brain, and it's suitable for this new lifestyle. But if you train the caterpillar to look for leaves on a particular color disc, what you find is that, and this is the work of Doug Blackiston, even before he joined my lab, he showed that the moth or butterfly actually remembers the original information. So the kind of more obvious thing here is that, wow, memory can persist across a total refactoring of the cognitive substrate, and that's amazing. And that challenges, for example, our computer technology. Our computer memories don't work that way. How do you keep the information while all the pieces are killed off, replaced, and shifted next to each other? But there's more to this, which is that the actual memories of the caterpillar are of no use to the butterfly. The butterfly doesn't crawl to the leaves. It doesn't care about leaves. It drinks nectar. You can't just map the memories directly. What you have to do is reinterpret them. You have to change them, because you have to adapt them to a completely new scenario, new ways of moving, new targets. Everything is new. What you've done here is abstract or generalize away from leaves to food, or maybe even beyond that, and reapply it to your new lifestyle. You don't inherit the actual memories, you reinterpret them as fits your new lifestyle. And so I won't talk about it now, but we can talk about it at the end if people have questions. There's another way to look at this, which is one way is that it's a passive process and all the work is done in the receiver. In other words, I get the memory and then I decide how I interpret it. And that's weird enough. But there's actually even a next stage to this, which is that if these memories are some kind of a persistent pattern within this cognitive system, are they purely passive? Or do they have a little bit of ability to do niche construction, like persistent and depressive thoughts that actually change brain structure to reinforce their own chances at repetition and survival? So there's this whole issue of some of this, looking at these memories as active data, not just a passive thing for the material to work over, but maybe some of these memories are actually taking an active part in making sure that they get remembered when they're translated into this completely new world. So let's talk about this translation business. I want to introduce the notion of bow tie nodes. And this is familiar to a lot of people who do networks. This is the most familiar form. There's a huge molecular network up here. Then there's some kind of hub node, some sort of, let's say, calcium signaling or something. Everything affects calcium signaling, and then calcium signaling affects everything else. Or you might have a gene regulatory network with some kind of hub node. The same is true of bioelectric. So last time I talked about bioelectricity and the idea that voltage is really this kind of hub, because lots of different ion movements impinge on the cell voltage, but also the cell voltage, once it changes, will kickstart lots of different downstream cascades. So again, you have this like center node, and the same thing is true in biomechanical networks. So what's interesting here is that one can look at these central nodes as communication interfaces, and this will become more important in the next couple of slides, because whatever is going on down here, you have to squeeze it down into a very compressed representation in a way that's going to be interpretable by all of this. So for example, in the case of calcium, it's not the case that everything up here triggers the exact same cascade down here, but calcium is informationally, it's quite compressed. And so you have to compress and generalize. So these middle nodes here are in effect translators. They're not just passive or static kinds of structures. They're actually communication interfaces. I think that's an interesting way to think about them. There are many other such communication interfaces. For example, money is such that it's a system that can allow very different other systems with different goals and whatever to actually communicate through a single scalar, a quantity of money. You can think about scientific papers as this, right? So you have lots of interesting ideas and brain states, and you squash them down into a published paper in a particular format, and then somebody will read it and have some of their own ideas, maybe some of yours, maybe not. And a particular kind of translator that we're very interested in is what I call the anatomical compiler. So the idea that someday in the future, you should be able to draw the plant or animal that you want, not at the level of molecular details, but actually at the level of anatomy. So organs, biobots, plants, animals, whatever. And this thing should be able to compile that description down to a set of stimuli that would be given to cells to get them to build exactly this. But the reason that I bring this up in this context is because what this is not is it is not a 3D printer. The point isn't to micromanage the behavior of individual cells or even genes. It's a communications device. It's a narrow interface that takes your design spec as an engineer or a worker in regenerative medicine and converts it into signals that will cause a collective of cells to adopt this as their target morphology representation, the pattern to which they build, to which they repair, the pattern that causes them to stop once it's done, right? It is supposed to be that kind of translator node that takes your goals and translates them into a biophysical representation for the cells to build. Okay, so now we have this. Now we have this concept of this kind of translator, this thin translator interface here at the beginning. And this is, by the way, this is the paper that all of this comes from. And there are lots of examples of this kind of thing that I'll share the slides later. I won't take the time to go through all that. But here's where things get really interesting. And we're going to use the same bow tie node idea. As a persistent active agent, you don't have access to the past. What you have access to are the engrams, the memory traces that the past has left in your memory medium, that being your brain, maybe your body if you're a planarian, or maybe if all of us. But that's what you have access to. You have access to the memory structures that were created. Now, that means that at any given point, so these are you can imagine the bread loaf of special relativity, and each one of these slices is, you can call it a selflet. It's a slice of yourself at any one time, the thickness, 200, 300 milliseconds, maybe something like that. So at each point in time, you have to interpret or reinterpret those memory traces. Again, because you don't have access to what actually happened, all you have to go by is the current material and energy patterns in your body. You have to reinterpret those patterns. And this is where we meet this idea, the paradox of change. So unfortunately, I forget who said this, but there's a classical thinker who said this idea that it was in the context of evolution. And it was the idea that if you're a species and you fail to change, you will probably die out and disappear when the environment changes. But likewise, if you do change and adapt in ways that match drastically different environments, then you're gone. Then also you're gone. You're not the same. And again, you've disappeared. So this issue that you actually cannot persist while remaining the same, and that what you have to do is commit to continuous change. And that's the only sense in which you're going to persist over long periods of time. And so here we're going to pull together these two ideas on an evolutionary timescale and on a cognitive timescale of a single agent. So imagine this again, visualize this as the bow tie. This is basically an autoencoder architecture that's used in machine learning. What you've got here is input data. So these are rich experiences. They're instances of things that you've observed. But down here, there's this bottleneck so this is all the past. It forms memories. When you form a memory, you don't just store all the pixels you've ever seen. You have to generalize. You have to throw away a lot of the correlations. You have to abstract the details. And what you hope, if you're a good learner, what you hope is that you've abstracted the generative rule. You've abstracted what do these past experiences have in common that I can remember. And then the past you has done this. It's formed engrams that are stored in your brain and body. Now future you at every given point has to decode them. So this, in a sense, so as you're marching along this 4D kind of space, this is the past, this is your current moment, the engrams that exist, and future you is going to have to decode them. So that means that one way to think about this is as memories as messages from your past self. So all of this is, in this paradigm, all of this is message passing. So of course, there's horizontal message passing to other agents at the same time, but there's also vertical message passing because your memories are messages left over from a past instance of you, and everything you do now is setting up future messages and bending the landscape for future you. So we can use some of the same conceptual approaches to think about memory as we do about communication with others. If we realize that every instance of accessing your memories is really an interpretation of information that was left for you. It's not, you don't have direct primary access to the past. So once you have that, we can then say, and other people have said versions of this. For example, here's Freud saying that the material in memory traces is subjected to rearrangement in accordance to fresh circumstances, right? And there's lots of work on confabulation and so on. So now this starts to make sense. So here's what's happened. During the encoding, this part, this is the intelligence part. This is the part where you're good at taking instances and figuring out what they have in common. So pattern abstraction. And it's I no longer think it's purely deductive, but it's largely deductive. And so you do this compression. And because you've abstracted a general rule from a bunch of instances, you now have compressed to a very compact representation. So here's your memory engram. And so that's why it has to be much smaller than these to force that compression. But then future you has an interesting option, and in fact, a requirement, which is that because you don't know what the actual details here were, all you have is the rule, you now have to decode this. And this is no longer a deductive process. This is inductive. This is creative. In other words, you're given these memories or memory traces, and you have to figure out what they mean in your current context. So the engrams are formed by the caterpillar, but the butterfly doesn't have access to the ground truth of what this was. He has to reinterpret this. And because you don't know what they were originally, you don't have any allegiance to interpreting them in the same way that they were interpreted before. You are free to interpret them however you want. And so that's true with any kind of communications act, whether vertically or horizontally. Anyone that gives you a message, when somebody says something to you that communicates a piece of information, you are not trying to micromanage your neurons to be in exactly the same brain state that they were in. The whole point of language and communication, signs and symbols and so on, is that it's a compressed, squeezed representation. You can then reinflate it onto your architecture, which might be quite different from the original. Even if you're both humans, your brains are going to be different. You can arrange it however you want. So this idea, already quite obvious to Freud and others based on clinical data, is the idea that all memories are reinterpretations. In fact, they're not even purely they're not non-destructive reads, as you would say in computer science. You modify them every time you access them, you modify them. And the whole thing becomes this very, very dynamic process. It's not about storing the memories with fidelity. It's about reinterpreting them for saliency and for adaptive usage. Now, this is all a story of conventional, like, behavioral memory on the scale of a single organism, but it works similarly on the evolutionary scale. So the reason that life is so good at dealing with these crazy new scenarios, some of which I told you about before, is that much like all of us, it cannot take past information literally, because you don't know the information you get from your genome. You don't actually know what it meant and what the right way to interpret it was in past environments. Chances are it's close, but nevertheless, you have to reinterpret it as best you can on the fly. And I think even during normal development, it's boring and consistent, because under normal circumstances, it does the same interpretation with the same data, with the same environment. But it doesn't have to be that way. And it doesn't have to be that way precisely because evolution does not make systems that are very good at fidelity and just hanging on to the same interpretation, but it makes these kind of play the hand you're dealt systems that have to figure it out from scratch each time. And so let me just give you two quick examples. This is a cross section through a kidney tubule in a newt. And normally there's, 8 to 10 cells that are making this thing up. Now, one trick you can do is you can force the proliferation of the nuclei before the cell gets to split early on, and that makes the cells much bigger. So the cell size adjusts to the amount of DNA. And if you do that, the cells get much bigger. The newt stays exactly the same. So that's weird. How did it do that? Well, if you take a cross section through the tubule, that now, bigger cells, but fewer of them make the exact same structure. Okay, so the number of cells adjusts to the new size of cell in order to achieve the same anatomical outcome. Again, that theme of achieving the same outcome when things change. The real kicker happens when you make the cells truly gigantic. And these are, I think, 6N newts. And then one enormous cell will bend around itself, leaving a space in the middle and solving the same problem in a completely different way using very different molecular mechanisms. This is cell-to-cell communication and tubulogenesis. This is cytoskeletal bending of some type. So not only do you have, again, the issue of problem solving and intelligence here, you've used different molecular mechanisms to solve a problem, but think about what it's like to be a newt, or in fact, any of us coming into this world. You can't count on having the right number of genes, the right copy number of your genetic material. You can't count on the number or the size of cells that you're going to have. So not just the environment changes, but your own parts. You can't even count on your own parts being the same. And you have to be able to achieve your goal even when your own parts are unreliable. And this idea that biology deals with inevitably with an unreliable medium, I think underlies all of these capacities. The reason that why would a newt be able to do this? This isn't something that happens to them in nature, but it does, because it's not just damage from the environment, but mutation on the level of an evolutionary lineage. Everything is going to change. Nothing is reliable. Your own parts are going to mutate, not just the environment. Everything is going to change. So I think, with possibly some exceptions like nematodes, but generally, I think what evolution makes are not solutions to specific problems. It makes problem-solving agents that do not assume much and that try to solve the problem from scratch every single time. This is a plant gall growing on this leaf. If not for a non-human bioengineer, which is, this one comes from a thrip. It's a little insect that puts some chemicals onto this leaf that serves as a kind of prompt to get these cells to build this crazy thing. If not for that, we would have never known that these cells were actually capable of doing that, of building this. The capacities of this medium are not obvious from the standard thing it does under standard circumstances, but if you start to perturb it a little bit, you find out that is extremely plastic. And I think the reason it's so plastic is because from the start, biology leans into this idea that everything is going to change and you can't take your information literally. And that's the confabulation aspect of it, that what you do, much like what human cognitive systems do, what the biology does is concoct a plausible, adaptive, useful story, let's say in anatomical space and transcriptional space, a useful story, not necessarily try to reconstruct the story that worked in the past. The summary of what I told you so far is that biological systems at multiple scales, all the way from molecular networks on up, are agents that operate in diverse spaces. They traverse and navigate spaces like anatomical morphospace, transcriptional space, and so on. I think that biology really commits to saliency, not overtraining on the meaning of past information, but assuming from the start that the medium is going to be unreliable and you are going to need to make sense of things from scratch every single time. And I think we can see some really interesting symmetries between agent scale cognition and the way learning and memory and so on work at the level of individual agents, and at the much bigger time and space agents in terms of a population scale evolution. And so I think fundamentally that this kind of problem-solving intelligence involving creative confabulation and having to use that kind of creative interpretation on the right side of that bow tie is there right from the beginning. Now, this has many implications. It has implications for computer science because our computer technology works exactly the opposite way. The reason that we have Moore's law and the difficulty of scaling up computer chips to have more and more computation on them is because if you make them too small, the quantum effects take over, the bits are unreliable, and you don't want that. You want your data, when you write a bit of data to the hardware, you want it to stay exactly how it is. You can't have the bits float. And in fact, computational architectures are very good at hiding all the bottom level details because you are supposed to trust them. When you write high level code, you don't worry that your bits might randomly change or that the metal underneath is going to do something weird. Every layer abstracts the layer below it and basically locks it down so that everything is extremely reliable, except for the field of unreliable computing, which is, I think, actually what biology is doing. And that's why in these computer architectures, you generally only get intelligence, if anywhere, you get it at the top level. Everything else is pretty dumb underneath. The parts aren't doing anything too terribly intelligent, but you hope that at the high level, it's doing something interesting. Biology isn't doing this at all. It doesn't worry about the keeping the information exactly stable. It assumes that it's all going to change. And we are now working on some architectures that take advantage of that biology. That's what I think really bio-inspired computing is going to do. But it also has applications for biomedicine, and that is what I want to talk about next. So the last time I was here, I talked about this kind of thing. So here are the kind of conventional approaches to medicine. So these are bottom up in the sense that they target the hardware. All of this is very specifically trying to micromanage the hardware. And what we've been working on are some top-down interventions, the larger class of morphoceuticals, which is this idea of conveying novel set points to anatomical homeostasis mechanisms, or getting the cells to build what you want them to build. And in particular, electroceuticals, which are different approaches to use the bioelectrical interface to get the cells to do something else. Just to remember that in neuroscience, the bioelectrical interface is what allows you to, for example, train a dog or a horse or communicate with another human without knowing any neuroscience whatsoever. You are not trying to manage their neuronal states. That interface, that center of that bow tie, is really good at taking information between two beings and remapping it from the sender to the receiver. And we've been using the bioelectric interface to tell cells what to do without micromanaging them. This is one example. What we've done is inject a specific kind of potassium channel RNA into some cells that are going to give rise to being a gut, cells that are fated to be a gut. And what we've done, the reason we've done this is to insert a particular bioelectrical pattern that we know from other studies encodes the meaning of build an eye here. That's the meaning of that pattern. The rest of the cells interpret that message, and this is what they do. So if you if you section these eyes, lens, retina, optic nerve, all that good stuff, and you learn a few interesting things. First of all, you learn that this bioelectrical interface is instructive, because you can use it to call up organs to gain of function effects in morphogenesis. The second thing is that it's incredibly modular. So we provided a very low information content signal. We did not have to tell the cells what genes to turn on or how many cells of each type there must be in an eye, or the stem cell regulation. We didn't have to do any of that. All we said is build an eye here. So it's a very high level subroutine call, basically. It also reveals higher competency than is otherwise available. For example, if you look at Developmental Biology textbook, what it will say is that the cells up here are the only cells in the frog embryo competent to be eye. And the reason they say that is because if you test this with a PAX6 so-called master eye gene, then yeah, the only way you get ectopic eyes is if you inject the PAX6 here. But what we found here is that's not because the competency of the cells is low. That's because our competency wasn't high enough in using the wrong prompt. PAX6 is not the best way to do this. The best way to do this is this voltage signal, in which case, actually, you find out that almost every region in the body is competent to make an eye. We just weren't communicating with it the right way. So this is, to me, a general issue about the field of diverse intelligence. When you're making assessments of some systems' competencies, you're taking an IQ test yourself, right? In terms of what have you figured out that it can do and how to communicate with it. And the final thing I want to mention is this. This is an ectopic lens sitting in the tail of a tadpole somewhere. What you'll notice is the blue cells are the ones we injected with our ion channel, but all these other cells were never touched directly by us. What's happened here is that these blue cells, there's not enough of them to build a proper lens. And so what they do is some kind of a secondary instruction. We tell the blue cells make an eye. They tell the others, you have to help. It's going to take this many to build, and they recruit them. So we behavior shape the blue cells. They in turn hack their neighbors to get them to participate. And there's lots of collective intelligences that do this. For example, ants will recruit nestmates to move heavy objects and so on. I've showed you before that this kind of approach, using this bioelectrical interface to signal for complex phenotypes that we don't know how to micromanage, and I don't think we want to micromanage them. Here's an example of our leg regeneration application, where frogs, unlike salamanders, normally do not regenerate their legs, but with a treatment with an ionophore, you can kickstart. By 45 days, you've got some toes, you got a toenail, and eventually a pretty good leg that's touch sensitive and motile. The coolest thing about this, to me, is that the treatment itself takes 24 hours. In an adult frog, the outcome of growing that leg takes a year and a half. During that time, we don't touch it at all. This is not at all about micromanaging the cells. It's not about scaffolds or factors or any of that. It's about convincing these cells on day one that they need to start traveling towards the leg regenerating path in morphospace, not the scarring path. And then that's it. Then you take your hands off the wheel and they do this themselves. And so again, this issue of modularity and low information content triggers inducing high complexity outcomes. You can do the same thing for the repair of birth defects. So here's a frog brain. You get to forebrain, midbrain, hindbrain in bilateral, this bilateral architecture. And what you can do is you can mutate a gene called Notch and introduce this dominant form. What you'll see is that the brain is completely screwed up. There is no forebrain. The midbrain and hindbrain are a big bubble. These animals have no behavior. They just lay there doing nothing. It's really a terrible defect. But it turns out that through our computational model, where we ask the question of, I showed you all this last time, so I'm not going all through it, but the borders of this organ and the structure of this organ are set by a bioelectrical pattern that is normally here, that gets completely screwed up when you mutate Notch, or when you hit them with alcohol or nicotine or any of that. And what our computational model was able to do is to tell us which channel, in this case HCN2, that we can target to restore the pattern back to normal, despite the presence of this aberrant Notch signaling. Here they are. This animal has the exact same mutant form, but he's got a good brain, and the behavior, the learning rate is basically indistinguishable from controls. And you can fix this either genetically by putting in new HCN2, or pharmacologically by opening. There's a couple of anti-epileptic drugs that open HCN2 channels, and that's good enough. And you can repair brain, heart, and face this way. And the last thing that I want to show is the cancer applications, which, again, what we were able to do is throw human oncogenes into these tadpoles. They normally make a tumor. You can predict where these tumors will form by their aberrant electrical map, because the first thing that oncogenes do is cause the cells to disconnect electrically from their environment. And at that point, basically, their cognitive light cone, or the size of their goals, shrinks drastically. So the border between self and outside world shrinks. And whereas before they were working on these very grandiose construction projects of muscle and skin and everything else, at this point, they're just amoebas. And the rest of the animal is just outside environment to them. And that's where you get metastasis, when they crawl off in search of a better life. So it turns out that what you can do then is you can introduce very nasty oncogenes, p53 mutations, KRAS, and normally they'll make a tumor like this, except that if you also use an ion channel to prevent the cells from disconnecting, so you force them to a particular electrical state, the cells don't die, the oncogene doesn't disappear. Here's labeled in red. It's blazingly expressed. It's all over the place, right? You should have tumors everywhere. This is the same animal. There is no tumor, because if you force the cells to connect, then they continue being part of the network and working on these morphogenetic goals. So at least what this means, these last couple of examples that I've showed you, what that means is that at least some problems, I'm not saying all, but some problems that are fundamentally hardware problems, Notch mutations, p53 mutations, KRAS mutants, some hardware problems can be solved in software. And understanding the signals that you have to give to these cells is the path forward, and that we're taking for limb regeneration, for birth defect repair, and cancer reprogramming. The last bit is this. So what I want to focus on now is this. It's in using these bioelectric interventions, we're clearly targeting a higher level control system than typical molecular medicine or pharmacology, because we're not trying to turn genes on and off. We're not talking even to the individual cells. We're sending a message to the anatomical collective. But even that ultimately, even that interface, I think we could do better. And that those things are described in more detail here. And also here, this is a brand new thing. You can take a look at that. But the idea is this, that if we take seriously the notion that what we have in these cells and tissues are problem-solving agents, then future medicine might look more like a kind of somatic psychiatry than it looks like chemistry, where what we're trying to do is manage the memories, the safety estimates, meaning how safe is the environment versus how hostile, the preferences, and the scale of the different agents within the body. And these are some of the things that we're doing. I'm going to just show you two very quickly. The first I call agential interventions, and we could talk about anthrobots, and then this crazy thing that's pretty new, and I thought I would try this out on you. So if I show you this and I ask you to guess what this is, it looks like some sort of a primitive organism that we found on the bottom of a pond somewhere. And I could tell you that we sequenced the genome, and the genome is 100% Homo sapiens. So this is the human version of the xenobots, which I showed you before. These are anthrobots. They are made from cells from adult, often elderly human patients. They donate tracheal biopsies, and then we buy them from a company, and we have a protocol in which these cells reboot their multicellularity, and they become these little motile proto-organisms. They have some cool features. This is what they look like. This is the protocol, but they develop, and they're covered by the cilia, which is what allows them to move. They have some really interesting features. One feature is that if you plate a dish of human neurons, so these are iPS-derived human neurons, and you take a scalpel and you put a big scratch through them. So this is a scratch wound assay. These bots are able to navigate the wound. But sometimes what they'll do is they will land at a particular region and they will assemble. And I will show you in a minute what happens. But they have a couple of others. So I've shown you the motility. They have a couple of other interesting properties. One is that they can self-heal. So here it is. We took a bite out of this one. We stuck it with a needle and took a bite out of it. Within minutes, this is probably less than 20 minutes all together, it heals itself. Also, here's an amazing thing. So these things are made of patient cells. They are totally wild type genetically. We have not edited the genome. There are no weird nanomaterials. There are no Yamanaka factors. There's no reprogramming, nothing like that. But if you actually look at their transcriptomes, thousands and thousands of new transcripts that their native cell source does not express. Both up and down, their transcriptome is completely remodeled by, and there's some, I don't have time today, but there's a paper that's going to be out at some point this fall, going through what some of these things are. But the point is massive transcriptional rewiring only because they have a new lifestyle. That's the only thing that's happened to these cells, is that they now have a different lifestyle. There's nothing wrong with their genome, but the lifestyle drives a huge change in the genes that they're going to express. And there's some very interesting aspects of what these things look like. And we're going to talk about that later. But the idea is that when they do these bots, we call these super bots or clusters. This is that neuronal sheet, and this is the wound. If multiple ones, so we know they heal themselves, we know that they have these very altered transcriptional programs. And then here's one of their capabilities. When a bunch of them kind of land in this area, what they do is they start knitting together the two sides of the gap. And if you lift them up 4 days later, this is basically what they do is they sit there and they try to connect this region. Now, who would have thought that your tracheal cells, which sit there quietly in your airway, have the ability to form a self-motile, novel kind of a construct that also has the ability to heal what looks like peripheral innervation. I should say also that this is like the first thing we tried. This isn't test 83 out of 200 things we tried. This is the first thing we tried. So my guess is that they probably have lots of interesting kinds of repair and other signaling capacities that we haven't even scratched the surface of. If we look at the list of different types of interventions in biomedicine, you have conventional kinds of things that target a specific state. Maybe it's a specific receptor. Maybe it's a specific.
Just to note that we're at time. Just
okay. Yep.
Like it's pretty kind of if you want to run it, but just
that's it. I got one more slide, and then we're good. Yeah. So what I'm interested in is the use of these biobots as, in fact, patient-specific biobots that don't require immunosuppression or whatever, that can be used as a very high-level intervention, because we don't know exactly where they are on the scale, but they may be able to do interesting context-sensitive things within the body that are really hard for us to micromanage. Okay, so let's see. Sorry, I couldn't see the clock because of this whole screen sharing thing. Like literally, we're out of time. I should just stop. Yeah. Okay.
We're now at 1 minute past the hour, or past the seminar hour. You have no idea how many questions there are in the chat right now. We have at least 10.
We could stay on for a little bit. If you have some time to answer questions, we could stay on, but also want to be respectful of your time.
Yeah. Why don't we, let's do 5 minutes of questions, and then I'll come back and do the rest of the talk later.
Okay. The first one up is Ben M.
Okay.
Ben Moskovich.
Great. Hit it, Ben.
Hey, yeah, thanks so much for the talk. Mine was actually on the first thing that you mentioned, and I want to let other people get to questions from later things too. But just quickly, you mentioning the barrier adapted heads on the planaria. There's like a ton of different genes to up or down regulate and how to do it consistently. So I was wondering if they turn on, like up and down regulate the same genes, and if they consistently and reliably do it in the same way, or it's like different approaches but same outcome.
Yeah, that's a super question. The original paper, we don't know because we basically just grouped all the worms together. But right now, as we speak, I have a data set sitting at the sequencing facility from individual animals. So we will be able to say probably in a couple of weeks, I'll be able to give you an answer.
Any hypothesis on that?
My hypothesis is that they're going to take different paths. For reasons that we can talk about separately, but I think they're all going to take different paths to solutions.
Okay, I hope so. That'd be cool.
Yeah.
Okay, great. Anna is next.
Yeah, so obvious question to me here is the animals that you see this in working this seems to unclear to me what kinds of scales that you're tending to see these kinds of agential intelligences, and that to me feels like a really big question for this like broader thing that people are interested in, that of understanding how biology translates and operates across scales. So do you think there might be like limiting factors to the scale at which these collective intelligences can operate for any reason?
In terms of the scale of the actual in terms of the scale of the material, no, I think these could be enormous. I've thought about at one point trying to design like a gravitational synapse that's the size of a solar system that would have the historicity in the dynamics of the planetary motions. I don't think there's actually a scale there. But if you're asking about what is the scale of the actual intelligence, how high can it go? I have no idea. My suspicion is that there is no limit. I certainly don't think we're at the top of the limit. I think it goes much higher, but I don't really have any good data on that.
Thank you.
Awesome. So next one is Siddhesh.
Hello, Professor Levin. Thank you for doing this. So I had two questions, but I'll just ask one. I remember like in one of the podcasts, you had mentioned that the planaria shape is stored in the bioelectrical network. So I have a question like where this bioelectrical network is stored, and when the planaria is cut into two pieces, like how this gets propagated to a different.
Okay, so the question of where does it come from is an interesting and deep question. We have to adjust our understanding of the question itself, because in the one hand, there is no. If you think about what kind of answer would make you happy, right? In pretty much nowhere in science is there a good answer to where does it come from, because whatever mechanism I tell you, you're going to say, okay, but where does that come from? And rightly so. You can track this all the way back. And for example, in the frog embryo, we can track it back to the single cell state, meaning the egg even before it gets fertilized. And so the question is, to me, the question really is, where does it come from? Means what is the most convenient information structure that I would modify to make changes, right? Where can we access the most efficient encoding? I think that in the end, much like with a lot of systems in biology and physics too, it's all going to come down to math. And what I mean by that is, here's a simple example, a Galton board. So you got a board, bang a bunch of nails into it. You can buy a toy of this for $20 on Amazon. And you take some marbles and you dump it in the top and the marbles go boom. And then you see this beautiful, if you have enough marbles, you see this beautiful bell curve. And you can ask the question, where does the Gaussian shape come from? It's not, is it encoded in the material of the marbles? Was it in the nails? Is it in the board? You're not going to find it anywhere there. So you end up with kind of everything eventually ends up as being a component of three things. There is the genetics, which gives you the hardware. So that's just the fact of having nails and marbles and wood and so on. Then there's the environment, which is whatever is going on at the time that impacts on the shape. And then there is wherever it is that mathematics come from. So eventually, if you really want to understand where that shape comes from, it's wherever it is that the truths of mathematics come from. And I think because of these things, because of these issues and questions about where do the xenobots form and function come from? There's never been any xenobots. There's never been selection to be a good xenobot or to be a good anthrobot. Where do their shapes come from? Why out of the box do they have these capacities? I'm more and more pushed to a kind of Platonic view of mathematics where there is an enormous, probably infinite space of possible forms. And some of those forms are boring things like triangles and whatnot. And some of them are probably much more complex and interesting. And when we build bodies or robots or software algorithms, what we're doing is we're building interfaces to this latent space. And I think that's the kind of thing we really need. I know it's not satisfying, but I really think we have to readjust the question, because there is no conventionally satisfying answer to where anything comes from, because eventually it ends up in where does mathematics come from.
Thank you so much for this perspective.
Thanks.
Great. So next one up is Andy.
Great talk. I was curious, at the bottleneck point of these networks in your computational models, are you specifically looking at quantity changes, or do you think that there might be something more complex in the kind of encoding of information and post-translational modifications? And are you tracking that at all?
Yeah, we look at this much wider than just, okay, so I definitely don't think that post-translational modifications would explain the kinds of things we see. Their channels in particular, the quantity doesn't mean much because they open and close as a function of physiological events. There's all kinds of interesting things about both the distributed spatiality of it, meaning it's not just a single cell voltage state, it's a distributed spatial pattern, but also there's biomechanical components, there are resonances and vibrations, and there's all kinds of stuff. It's not just post-translational changes.
Thank you. So next one up is Vadim.
Yeah, this is a very inspiring talk. Thank you very
Hey,
much.
Vadim. Thank you so much.
The bow-tie model resembles the evolutionary hourglass model, with the ideal phylotypic state presenting the most concerned morphological space.
Yep.
I just wonder if you could comment on that and what this means in terms of information transfer, latent space representation in this model.
Yeah, I think it's connected. I have a paper with Eva Jablonka where we a little bit make the connection to the phylotypic stage. I haven't worked this all out. What I do think is that the conventional models of compression of information and the kind of the Shannon view and all of that, where you literally want to understand the fidelity of the information along a medium, right? And you want to like, how much information can we pack in, all that stuff. I think this is a different angle on it, because I think much of the work gets done on the receiver end in terms of this creative interpretation. And so I think we have to ask, when we have these phylotypic stages and these other things, I think one question we want to ask is, what about that stage makes it easy for the future to reinterpret the past? I think that's one way we have to start looking at it, is that not just the mechanical, okay, it's easier to canalize to this or whatever, but actually, what is it here that facilitates this? Because the big gap here that I haven't filled yet, and we're trying, but that capacity to actively reinterpret past data in new ways, that's not a simple thing. We don't know how that works. That's a huge gap. And I have a feeling that a lot of the networks that we see are shaped precisely to make that easier. That's why they're shaped like that. But because we don't know exactly how that works yet, I can't tell you exactly what it is about the phylotypic stage that does it. But I think that's how we have to start thinking about it.
Thanks so much. Okay, so I think the last question we have time for today is Creon.
Oh, hey, Michael, thanks. A delightful and informative, inspiring talk as usual.
Awesome. Thank you so much.
Yeah, I have so much I'd love to talk to you about, but I'm just going to ask you this one thing. I suspect the answer is yes. Have you seen the paper, The Genomic Code, the Genome Instantiates a Generative Model of the Organism by Mitchell and Cheney?
I have.
You have?
I have, yeah. So this is an idea. Yeah, I have, and I'm glad other people are on it. This is Giovanni Pizzullo and I put forward something like this in 2016. This entropy paper that I pointed you to has that concept as well. Yeah, I definitely think this is onto something. Yeah, I think it's important.
Yeah, just to mention something maybe to the whole group here briefly, is that in that paper, which has the autoencoder model that you also showed in one of your earlier slides, they specifically hypothesized that the encoder side is evolution. And then you have the latent representation, which is the genome in their model. And then the decoder side is development.
Yep. I think that's perfectly reasonable. That's certainly one way to look at it. What I think is fundamental to my view is, and this is a polycomputing kind of approach that Josh Bongard and I developed, where I don't think there's one correct way to look at any of this. I think there are observers in the physics sense all over these biological systems, and depending on which one's perspective you take, the different mappings start to make sense. So I'm fine with their interpretation of it.
Yeah. And in particular, the idea that development is the decoder, which turns the genome into the organism type, whatever. Like that's compressing a lot and leaving a lot out. Like development, there's all kinds of loops and nested structures. Yeah. Done.
Sure. Yeah. And if anybody's interested in that stuff, you can drop me an email and I'll send you a couple papers that Zullo and I wrote, and also Daniel Lobo, talking about this very idea, the compression and decompression of development and what it means for basal intelligence.
Thank you so much, Michael. We really appreciate you coming on. It was such a pleasure. And
Thank you so much.
everyone for joining. Thank you.
All right. Thanks, everyone. Be
Great.
well.
Have a great rest of your day.